coast of Svalbard in the Barents Sea. And that's a very fascinating point, because from the south, water from the North Atlantic is moving northwards, and from the north, Arctic water is moving southwards. So right here they are coming together, and so there's a sharp boundary between very warm water coming from the south and very cold water coming from the north, and that's what we call the frontal zone.
My name is Eva Loy.
Lorter Sem.
Einat Sandbank.
My name is Njal Heddle.
My name is Frida Knossen.
I'm a senior researcher working at Aquaplan Niva, and my background is marine biology, more specifically algal ecophysiology.
I'm in Tromsø for an internship for the ending of my degree, and I got the opportunity to join this cruise. I don't have a specific task or anything. Getting the samples in, processing the samples, and just preparing for the data analysis and data collection, actually.
I'm a PhD student from Memorial University. I was hired as part of the Polar Front project. My role is to help study the fish and zooplankton diversity biomass using acoustics and nets.
So I'm a biological oceanographer, so one of my roles is to describe the physical environment. But apart from that, I'm looking also at the zooplankton community, together with Eva, and looking at the productivity of the system as well.
I'm a marine biologist and also a science illustrator. I started last year with my master thesis on the Polar Front project, and also studying fish diet and macrozooplankton distribution across the front.
The scientists here on board are actually all coming from different institutions. We have colleagues from Aquaplan, and we have colleagues from the university here in Tromsø. It's a bit a special day because it's the first day where we start sampling, and this is always a lot of excitement. You know, there's so much preparation that goes into one of these cruises. It starts like maybe 4 years ago when you write the research proposal, and then it's a very selective process, and we were selected for funding. And so we had our first cruise last year. So we have basically a biological team here on board that does everything from microbes to vertebrates. And the major research question for us is the area where the Arctic water and the polar water, where they are meeting, which we call the polar front, is that a very specific place that is favoring certain organisms and is important in their life cycles. And for that we need to come several times, so we have this opportunity to come 3 times.
The best part of my job, or my study so far, has been meeting all the different researchers, because they're interested in a very specific topic and they're always passionate about this stuff. hear them talk about the work they do, it's always a bit inspiring, and it like makes you view things from a different perspective, which is always really nice and interesting.
We're preparing for the station because when we get the fish and the zooplankton, we want to process them quickly. It takes a while. So we're trying to streamline it and make labels ahead of time, make sure we know our data sheets and where they are, what's on them, to make it more streamlined, because it's sometimes a little bit high stress, fast paced. You're dealing with live animals, you want to treat them kindly. And this is waterproof paper, so it will not be ruined when it goes in a bag with a fish. We'll still be able to read the label clearly.
Our main question in this project is to understand whether the polar front is really a region of increased productivity, and if so, whether this is locally induced or whether it's more due to advection of organisms and biotic energy from different water masses meeting in this area.
Here I'm filtering for chlorophyll, which is something that we use as an estimate for the amount of phytoplankton biomass present in the water column. We collect chlorophyll measurements from 6 different depths throughout the water column to see where the phytoplankton are most abundant and just how abundant they are. Chlorophyll is a pigment, just like the pigment that we find in grass. It's necessary for doing primary production. So because of that, because it's present in all phytoplankton, we can use it as an estimate when you measure the concentration of chlorophyll in the seawater, roughly how many phytoplankton are also present. It's not a perfect estimate because there are nuances to just how much chlorophyll is present in different types of phytoplankton or just how well they're growing, but in general it's a very easy and very common method of measuring phytoplankton abundance.
What we're doing right now is measuring an in situ profile of algal physiology, and this tells us in which state the algae are or whether they are experiencing any form of stress. And we are doing this right now in a profile where we lower an instrument and get a continuous profile of the in situ conditions. But in parallel at the stations, I'm also doing this with the same instrument in a bench setup, where I can measure under much more controlled conditions and also have light curves where I increase the light intensities gradually and also measure the physiological response to that.
So that's one of the instruments. So we call this rosette the CTD, conductivity, temperature, depth. When we lower them in the water column, then we get these profiles of salinity and of temperature along the entire water column. But there is actually a fluorometer on it as well. So that's a fluorescence measurement based. And then there is also a power sensor. So that's photosynthetic active radiation. So that is kind of the light that is available for those primary producers, so for the phytoplankton. Oxygen is also there, so that gives also a state of the system and the phytoplankton in particular. There are also these water bottles, the Niskin bottles, so then they, when we lower all of that into the water, then we can decide exactly at which depths we want to sample water. So we just press a button up in the instrument room, and then we can get water from 100 meter or 200 meter. And then when it comes back up on deck, we have all that water and can process it.
We're looking at the distribution of fish across the front. We have the boat's echo sounder, which comes from the surface right under the boat and projects the sound waves down into the water.
So here you have the depth. The total depth is 220 meters from the surface to the bottom. And each of these lines here is either one organism or the group of organisms, school of organisms.
They attenuate and you get more noise, and the signal you get back is not as clear. What we created or built was this Frankenstein collection of instruments, and one of the instruments that's on it is a smaller transceiver and a couple of transducers. And by lowering that, we hope to have a clearer picture of what's happening throughout the water column, because you're measuring over or recording over shorter distances, so there's less attenuation of the sound wave, and you can get into deeper depths with higher frequencies, which is very nice.
We are using glider technology in this project because these autonomous vehicles, where we can put a number of instruments on, have the big advantage that they can collect continuously data with a very high temporal spatial resolution that we cannot map in the same way from a vessel.
The sea glider has an EK80 echo sounder on it. It also has a CT sensor for connectivity and temperature, as well as biogeochemical sensors. It also in the nose has an UVP6 LP that counts particles or plankton if you want.
So with the big ship, we obviously stop every now and then and we take stations where we collect samples, but then it's a snapshot from one point, and the gliders give us the context. It's programmed to go undulating down and then up again and down again. And whenever it surfaces, it can communicate, and it can also be piloted actively by someone else.
We had an issue with the communication, mostly because Wi-Fi wasn't the best out there. We also got close to the side and just wanted a picture and knowing how it laid in water. And once they got that, they sent a command and then it dived. And it takes a couple of minutes for it to dive to feed out the oil in the bladder. So that's why it took so long time. Everything seems good, so we're gonna do this sailboy now.
The sailboy, which is the other autonomous vehicle we are going to use, is a surface vehicle.
The sailboy also has an EK80 echo sounder on it. It also has an echo triplet and a CT and a oxygenometer.
So that is floating on top of the surface and has instruments mounted, looking downwards and giving us information about what we have in the underlying water.
So in the Barents Sea we have 2 and maybe 3 or 4 main fish species that serve as an important role for transferring a lot of the energy from lower trophic levels, such as zooplankton, to many higher trophic levels, fish, seals, mammals, including some commercial fish species such as Atlantic cod, haddock. 2 of the main species we've been finding are capelin and polar cod. And so, as the name maybe suggests, polar cod is an Arctic species, whereas capelin is more a subarctic species. And so, the idea is that the polar front could serve as an important feeding ground for them. So they feed mostly on zooplankton. And in the Barents Sea, an important part of the zooplankton is attracted with the Atlantic current. And so it could perhaps accumulate at the polar front, and also a part of the zooplankton grows or is born in the Barents Sea. So yeah, that's why we're assessing if these fish are feeding here.
nets out, and then we get the nets, everything that's in the nets collected in this bucket, and this bucket is then taken downstairs into the lab for the processing. So we sort all the individuals, or like the species that we find, or the groups of species that we find, and this is then later used for analysis on stable isotopes, for example, or just dry weight or lipid content of the zooplankton, to see how much reserve they build up for the winter period, where of course like productivity is lower, so they have to prepare a little bit as well.
So we're using different nets to catch different types of the zooplankton community. Like the multi-net and the WP2 net, they have a mesh size of 180 micrometer and an opening of 25 square meters. And with that you capture copepods and smaller copepods. But then the larger like krill and amphipods, pelagic amphipods, they feel the net coming. So then you use nets with a larger opening and a wider mesh size. And that's why we use the WP3 net, which has an opening of 1 square meter and a mesh size of 1000 micrometer. And the hope is here that we catch, we are looking for colonists, for the females, for egg incubations. And by using that mesh size and that opening, we'll hope that we get rid of all the small stuff, so it's easier to sort. And then previously we used the tucker troll, so that's the one that actually, so all these nets that go out right now, they go vertically, so we just take them out and break them up vertically. But the tucker troll is actually trawling, so horizontal. And the idea is also that you catch the larger zooplankton species that are fast swimming and that can feel the net coming in the vertical ones. So that's why we have all these different types of nets.
Pelagic trawl, where a bunch of fish came in, same procedure, right? So the net goes out, collects a bunch of stuff. This is collected in a bucket, and then we sort them species wise, and then we do the measurements, so length, height and weight.
So we've been sorting the different fish and then dissecting the stomachs to later investigate for the stomach contents. So we've been catching a lot of jellyfish again, and also mainly capelin, and some polar cod in the Arctic waters. But the polar cod were often very small, and about this size maybe. And yeah, that's been the main things we've been catching. And a few other more subarctic species like Atlantic cod and some pollock as well. Compared to last year, we also found a lot of big capelin schools. Now it's been a little bit less, but for the polar cod, last year we found also many bigger ones, adults, and that were also feeding closer to the polar front. Currently, we've only found the smaller polar cod in the Arctic waters. Question mark, where are the bigger polar cod? Perhaps further north? We don't know yet.
The otter is actually doing kind of great. It's just the weather that's not the best right now. So I wanted to test it today, but it wasn't with us today. So maybe tomorrow. So the software is like really easy to use. So right now we're looking at the boat. I can just move this cursor on the other side, and then we'll turn around, just a moment. And then it goes to that point and just shows 4 nodes. That easy. Well, the other worked amazingly. We had a pretty good test run. I haven't looked through all the data yet, but it seems to have done well, actually.
Did you factor in how to get it out of the water once you put it in?
We had a slight idea, but more, no, it wasn't planned, not at all. But the crew are fantastic. So I suggested otter fishing, which they actually agreed upon. So yeah, we opened the CTD hatch, the crew took the crane over, then we drove the otter to the hatch, and just fished it out of the water. It was a bit of try and error, but it worked well.
I'm looking at the calamus copepods, so that are these little crustaceans. They're about the size, like 3-4 millimeters, like about the size of a rice corn. And they are, in terms of biomass, one of the most important zooplankton species in the Arctic and subarctic, so here in the Barents Sea where we are. And they're so important because, you see, they have this lipid sac here. So what they do in spring, they reproduce in spring, and then they use the entire summer to eat the algae. Basically, they spend the whole summer eating and trying to get fat, because in the winter time, at the end of the summer, they migrate down to deeper depths and overwinter. And during winter, they basically don't have any food. So they need those lipid reserves to survive the winter and to initiate reproduction in the next spring. So we're taking samples from the surface and from the deep water, and then we take pictures of each of a subsample of the individuals. We determine the stage, the life stage, and then we measure basically afterwards the lipid sac. And from that we get an estimate of the lipid content of the individuals and also of the fullness. And the idea is then that we get an idea of how much energy is in the system, if it's still most of it is in the surface or if they already migrate down, and how it is distributed across the front.
This was now our 9th station, and as you can see here on the whiteboard, these are all the devices that we have been using. We looked at phytoplankton, zooplankton, and in the very end we did fish trawls. So that was our standard program, going from bacteria up to fish at each of the stations. And we were successful at doing that at each of our 9 stations. So we had 9 major stations, and today at 76 degrees 19 was our final major station of the expedition. But the science is not completed, so we will continue. We have the MVP, that's not most valuable player. This is actually a device that is towed behind the ship while we are making our way towards Tromsø. And we had deployed that on the way north, now we will deploy it on the way south, and it gives us a really good picture about physical and biological properties in the uppermost 200 meters of the water. And this we will continue as far south as we can, and then at one point on Saturday, Captain will say, time over, now we need to steam to Tromsø. So we pick up the glider and we deployed that from the ship at the beginning of the cruise, and so today at about 11 o'clock we will recover. That means people will go out in a small boat, pick it up and bring it back to the vessel. Will be a bit of a challenge because it's a little bit more wavy and windy. We have now 11 meters per second wind speed, which is not horrible, but it's more than we had 2 days ago, where it was just 3 meters per second. So it will be a little bit more rougher ride for those in the boat, but it will all be fine. We have two major tasks we have to accomplish. These are my least favorable one, is to pack. So all the stuff we brought onto the ship, we have to put back in boxes so that it can be safely loaded back. So you need to bring each pencil, each microscope, each net, everything we need to bring.
Then there are some sensors that have been borrowed from other project partners who couldn't join the cruise. So some of the equipment will be sent back to Canada and some to Poland. So it's been made ready for shipping and needs to go through customs and things like that.
There is no shop where we can buy anything, so we really need to make sure that we have everything with us, and now we have to put everything back because at the very end we need to leave the ship as a clean ship. So that means we also have to swipe the floors, clean the tables, that the next science team that will come on board really start working as they should.
I think the cruise went very well. We were sampling 9 complete stations and then additional small stations. So that's more than what we accomplished on the previous cruises, but this cruise was also a little bit longer. The chlorophyll A concentrations were much lower than on the spring cruises, but this is to be expected since now we're talking about summertime and nutrients have been used up to a great extent. What was surprising to me is that in the photophysiology measurements I did, the photosynthetic yield, which is a so-called happiness indicator or indicator of potential stress for algae, was almost the same as on the spring cruises, which I would not necessarily have expected. But that tells me that supposedly these algae that we find now again, although it's a completely different community than we found in spring, is very well adapted to their current conditions and managed to overcome shortage of inorganic nutrients by other sources of taking them up.
Everyone was working incredibly hard, so I think we got really a lot of things done.
Biggest takeaway from this cruise, being my first one, has been just how a team works on such a cruise basically. So we're all here with the same aim or like overarching interest in this case. So that's something that I've enjoyed a lot. So just basically talking to everyone about what they found and how they did it and the procedures they used. So also like the different devices that were deployed, and I was really surprised by how many different devices were used, all these different perspectives on different topics and subjects, and that was really interesting to see, and also a little bit inspiring as well.
That's one thing I really like about these cruises. It's not only an important scientific experience, but it's also a cultural experience when people from different cultures merge.
Like a superstar.